to Interferon Preparation and Detection, Part 1. Creative Biomart. Introduction. In this paper, interferon-inducing agent was used to prepare human leukocyte interferon as an example to introduce the preparation method of interferon. Creative Biomart. Experimental principle. Steroids are a class of highly active, multifunctional proteins produced by interferon-inducing agents that act on biological cells. After it is produced and released from cells, it acts on other cells of the same species to obtain antivirus and anti-tumor immunity. Creative Biomart. Experimental principle. The so-called interferon inducer refers to a substance that can induce the production of interferon by biological cells. In practice, two methods are used to prepare interferon. The first is to use interferon inducer to induce the production of interferon in certain biological cells, which can be used after being extracted and purified and qualified. The cells used in this method are mostly peripheral blood leukocytes. The second method is carried out by genetic engineering, that is, the interferon gene is introduced into Escherichia coli, and interferon is produced by culturing Escherichia coli. At present, large-scale production of interferon mainly uses genetic engineering methods. Creative Biomart. Major equipment. Cell culture equipment such as culture flasks, porous culture plates, incubators, microscopes, rotary incubators, etc., water baths, etc. Creative Biomart. Experimental procedure. 1. Preparation of inducing agent, using NDVF attenuated strain, stored in chicken embryo allantoic fluid at -20°C. Creative Biomart. 2. Preparation of induced cells, sterile human peripheral blood, using human umbilical cord blood, or blood bank storage blood, placed in a sterile bottle containing heparin, stored at 4°C for no more than 24h. Induced cells, white blood cells, are not extracted separately and replaced with whole blood. Creative Biomart. 3. Preparation of crude interferon. 1. Add an inducer. Add 1 ml anticoagulant whole blood, plus 0.2 ml of inducing agent, that is NDVF allantoic fluid. The blood coagulation titer is not less than 1/640. 2. Warming adsorption. Anticoagulant whole blood with antibiotic was placed in a 37°C water bath for 1h, shaking once every 15 min, so that NDVF was adsorbed on white blood cells. Then centrifuge at 1000 R/min for 20 min, discard the supernatant, and leave a precipitate. Creative Biomart. 3. Preparation of crude interferon. 3. Incubation with nutrient solution. Add equal nutrient solution to the above precipitate according to the amount of anticoagulant whole blood 1 to 2 times. Mix well, and rotate in a temperature of 35 to 36°C for 18 to 20h. 4. Centrifugation and acid treatment. The above culture was centrifuged at 2000 R/min for 30 min. The supernatant was taken. The pH value was adjusted to 2.0 with 6 mol/L hydrochloric acid, and the NDV was inactivated in a refrigerator at 4°C for 5 days. 5. Neutralize. After acidification for 5 days, adjust the pH to 7.2-7.4 with 6 mol/L sodium hydroxide. Creative Biomart. To be continued. Collected by Creative Biomart. Creative Biomart. Contact information. Email, contact@creative-biomart.com. Website, www.creativebiomart.net. Address, 45-1 Ramsey Road, Shirley, New York 11967, USA.